Now we are live, and as I was just saying before, we are going to start session 4 about organisms and genomics. So let's start with Anna, who is going to talk about this fantastic little creature, marine anemone, and then Kelly is going to talk something slightly bigger, just slightly bigger, an urchin, and Camille, something a little bit even bigger, and I hope you're smiling, so it means that you can hear me. Yes, okay, we will talk about the behavior of octopus. I also introduced the jury members who are here, so thank you a lot for being with us again. So online, we have 2 people from here, but who are in Paris currently for a meeting. So Eric Rottinger, they cannot see you, but they can hear you, I hope.
Hello, why don't you see me?
Yes, we can hear you. And okay. So Eric Rottinger from Université Côte d'Azur and CNRS from the lab IRCAN. Aldin here at Daniel, also from IRCAN lab, research engineer, also working on Idarians. Stefano Ciotto, who is working at CNRS IMEV at Villefranche. Stefano, do you hear me?
Everybody? Yep.
Yes.
Yep.
All good. And Sarah Sergi, that of course you know as well. Okay. So you have the floor for the next 15 minutes.
Good afternoon, everyone. Thank you for being here. I'm really glad to be presenting to you my master's thesis internship that I carried out in the Scientific Center of Monaco for the last 6 months. The topic of my master's thesis was to study sea anemone Exaptasia pallidus NLR's function during a bacterial infection. So let me first begin by talking to you about the immune system. In vertebrates, it is twofold, and it consists of adaptive immunity and innate immunity. And the vertebrate innate immune system is relatively well characterized and understood in comparison to the immune system of invertebrates, who interestingly only possess innate immunity. Now, a very important defense mechanism of the immune system is the formation of inflammation. And in fact, in recent years, there has been an increase in the prevalence of inflammatory disorders worldwide. A gene family called the NLR gene family has been implicated in many of these diseases. NLRs are nucleotide-binding oligomerization domain-like receptors, which act as regulators for immune responses, as well as sensors for intracellular pumps and dumps. So a prototypical NLR protein consists of 3 parts. First, we have a C-terminal leucine-rich repeat domain for ligand binding. Next, we have a nucleotide binding domain, usually a NAC domain. And lastly, we have an N-terminal effector domain, which varies from NLR and is used to classify them into subfamilies. Some NLRs are also capable of oligomerizing into multi-protein complexes called inflammasomes, and inflammasomes have been implicated in many inflammatory disorders and also in certain cancers, making NLRs very interesting targets for treatment. Interestingly, though, the classical invertebrate model organisms for studying immunity do not express any NLRs, while in contrast, we have cnidarians, which include corals and sea anemones. They have shown to have very rich and diverse NLR gene families. Now, research on NLRs has been relatively slow. For example, of the 22 that we have identified in humans, only 10 have been characterized still. And this is the reason why the Ecosystems and Immunity Laboratory team of the CSM started a new research project a couple years ago, aiming to contribute NLR-relevant knowledge for the treatment of human inflammatory diseases. For their model organism, they have selected Exaptasia pallida, which is a sea anemone. I will call it Ectasia for this presentation. And in order to induce an innate immune reaction in the anemone, they selected marine zoonotic pathogen for humans, Vibrio parahemolyticus, or VP. Now, this methodology is still relatively new for the laboratory, and it still needs quite a bit of optimization, as well as control over the animals. So they first began the project with a transcriptomic analysis on the innate immune response of Aptasia to VP infection. And what they discovered from the RNA-seq data is that 78 sequences were found to be annotated as NLRs, of which 6 presented to have both leucine-rich repeat and NAC domains. The study was also looking for potential interacting caspases, as caspases are very important in the regulation of activating inflammatory signaling pathways. From the RNA-seq data, they found 29 sequences annotated to be caspases, of which 4 were highly conserved. Here are some of the results from the same study. We can see the regulation of 2 NLRs and 2 caspases over a 12-hour infection period. First, we have an NLRC5-like, which was shown to be upregulated from 6 and 12 hours post-infection, and an NLRC3-like, which was shown to be downregulated at 12 hours. Now, some potential interacting caspases could involve caspase 3, which is usually known as an effector caspase, which were shown to be upregulated throughout the entire infection period, and an caspase 8, which is usually known as an initiated caspase, and it was shown to be upregulated at 3 hours and 6 hours. So based on the current knowledge on this topic, as well as the results from this study, my team and I developed a hypothesis of how they might be interacting in aptasia. We hypothesized that pumps and dumps trigger an upregulation of aptasia's NLRs. We think that these NLRs first activate caspase 3, and caspase 3 activates caspase 8. The activated caspases then lead to the activation of programmed cell death pathways of apoptosis and pyroptosis. This leads me to the aim of my thesis, which was threefold. First, to optimize a colony-forming unit-specific BP protocol in order to control infections better. Next, to determine the survival curve for our model organism. And lastly, to analyze the gene expression of NLRC5, NLRC3, caspase 3, and caspase 8 in aptasia during BP infection. And the work was carried out with the hypothesis that NLR and caspase genes in aptasia are regulated in both the time and concentration-dependent manner. I carried out 3 different infection experiments in my thesis, and in order to control the infection well, first I had to optimize a CFU-specific BP culturing protocol, where we could reach infection conditions of 10 to the power of 8 colony-forming units of BP per milliliter in experimental conditions. I discovered that time, agitation, volume, and surface area are the factors that most affect BP growth. And using a combination of these 4, I built a CFU-specific BP protocol. Using this protocol, I cultured BP, I took the CFU of the BP stock, and simultaneously I infected the anemones. In one of the assays I carried out, I also exposed aptasia to dead BP, in which case the BP stock was killed at 65 degrees before inoculation. The anemones were incubated at 27 degrees with no light, and the water was changed to filtered seawater after 48 hours of infection exposure. After this point, the 3 assays diverged, and now we can look at them in more detail. So in the first assay, I questioned the effect of both live versus dead BP at 3 different concentrations in terms of aptasia's survivability to the pathogen. Here I took daily observations and photographs, and I also recorded the mortality. In the next assay, I looked at the amount of BP both inside and on top of the anemone, which was called the in-hospite BP, and the evolution over a 6-day infection period. Here I also looked at the concentration of BP in wells around the aptasia with aptasia present versus with no aptasia present. And in the last assay, I questioned the exposure period of BP. I questioned the effect of the VP exposure period on the gene expression of aptasia's NLRC3, NLRC5, caspase 3, and caspase 8. Here, after the exposure period was over, RNA extraction was carried out on the anemones, and reverse transcription, quantitative real-time PCR analyses followed. Here we have the photographs from the dead BP-treated anemones from the first assay at 3 different concentrations over an 8-day infection period. And what we observed is that mortality was only observed at the highest concentration, which therefore was the lethal concentration of BP. At this highest concentration, 5 distinct stages of infection could be visualized. First, the shriveling of tentacles, which you can see here very well. Next, mucus production was increased, and after which algae cells started to be released. You can see here, these are algae cells. or bundles of algae cells. And finally, the anemone detached itself and the body liquefied. So the photographs from both the dead VP and the live VP treated anemones were very similar. This is why I've only presented the dead VP treated anemones here. When we look at the Kaplan-Meier curves depicting the percent survival of Aptasia over the same 8-day infection period, I identified that 50% of individuals exposed to live VP had died by day 5. However, this threshold was passed on day 8 with dead VP exposure. Therefore, there was a 3-day delay in the lethality. The cause of death for the dead VP-treated anemones was also speculated to either be caused from the contact with the bacterial ligands from the dead VP, or possibly a secondary infection had evolved during the infection. From this assay, the take-home message is that the tolerance threshold of Aptasia to VP is between 10 to the 7th and 10 to the 8th CFU per milliliter. And this threshold was tested in the second assay, where I evaluated the in-hospit VP amounts per anemone over a 6-day infection at specific time points. What these results basically tell us is that there was very high variability between the biological replicates. This is largely due to the fact that each anemone had their own specific bacterial load, and each anemone passed to the next stage of infection at their own pace or timescale. When we look at the CFU concentration of the VP concentration in CFU per milliliter in the wells with both Aptasia present versus the wells with no Aptasia present, we can see that the presence of Aptasia decreases the amount of VP in suspension. This could be due to the fact that Aptasia is known to filter the water, therefore the VP ends up inside and not in the surroundings. So now that we've looked at how VP affects Aptasia on a morphological level, let's look at the gene expression analysis. So I looked at 2 different NLRs, NLRC5, which was upregulated in the RNA-seq data, and NLRC3, which was shown to be downregulated. To analyze gene expression, I used the Puffle method of quantifying gene expression relative to an untreated control and normalized using a stable housekeeping gene across conditions. Sadly, we could not statistically validify the results presented in the RNA-seq. However, the trends that we observed were similar to the ones exhibited in the transcriptomic assay. The high variability that was witnessed in the second assay between the biological replicates possibly translates to differences in gene expression, and therefore, in order to validate these results, we would need to use more biological replicates than just the 3 used here. When we look at the potential interacting caspases, our hypothesis was that NLRs first activate caspase 3, which then activate caspase 8. And my results show that there was a significant upregulation of caspase 3, both at 12 hours and 24 hours post-exposure. When we look at caspase 8, we saw that there was a significant upregulation at 24 hours post-exposure. Therefore, we could say that our hypothesis might in fact be correct due to the fact that caspase 3 is activated first and caspase 8 follows after. The results from this assay also show that caspase genes in Aptasia are regulated in fact in a time-dependent manner. To conclude, my results show that both dead and live VP are lethal to Aptasia at concentration 10 to the 8th CFU per milliliter. And the next step here would be to use the identified sublethal concentrations in order to prime the anemone first at those concentrations and reinfect them to evaluate their trained immunity potential. My results also point to the fact that there is a threshold of in-hospita bacteria which once reached, the anemone cannot survive the infection. And so in order to better understand how the infection evolves in Aptasia, it would be good to determine the in-hospit VP quantities per infection stage. Lastly, using all the information gathered from my report, the next step would be to infect anemones at both sublethal and lethal concentrations in order to evaluate their immune gene expression in order to evaluate their immune gene expression in terms of whether it is also concentration dependent. And the overall goal for the next step of this project is to assess the role of anemone NLRs in apoptosis and pyroptosis pathways in more detail. I would like to thank the Ecosystems and Immunity team for accepting me and for all of their help they've given me. And I would like to say a great big thank you to my great few supervisors who are actually here right now. Thank you, Francois, and thank you, Alexander. And thank you, everyone who came to listen. Please feel free to ask me any questions.
Thank you, Anna, for your presentation. We'll open the floor to questions. I think you're right. And we'll start with you, Aldin, if you agree. Aldin and then Sarah.
So thanks, Anna, for this beautiful presentation. So you mentioned that live and dead Vibrio have an effect on the life of the sea anemone. So can you comment on that? And is it using the same concentration?
Yes. So the concentrations in that assay were the exact same for the live and dead. And we made sure because that we used the same livestock to make the dead VP concentrations. So we took the CFU and then we killed it and then we inoculated. And so the concentrations were definitely the same. But the theory is that since it has a lot of bacterial ligands in the mix, and we know that, for example, a very high dose of LPS is already capable of initiating a very strong response in an animal. Therefore, it's the same. It's basically just giving a huge dose of bacterial ligands and it initiates a very strong response.
How long can you keep this dead Vibrio before attenuating the effect of this molecule? So on the sea anemone, do you know that? Can you keep? What would be another control that you can add in your experience regarding to go deeper into the effect of Vibrio on the infection of the sea anemone? Which kind of control?
I would also use a control which just has LPS, for example, to compare them to, because I feel like the dead VP has more, so it will initiate an even stronger response. But yeah,
Okay.
I think LPS.
So did you starve or fed your sea anemone during the experiment, or did you use, starve one, fed one? How was it?
So the anemones were separated from their containers 24 hours before infection. So they had time to get used to the wells and fix and yes. But after being separated into the 24 well plates, they were not fed, and they were not fed during the whole experiment. Okay. And during the whole experiment, they were kept in darkness. So no photosynthesis was happening either.
Do you know, or did you test if you change the feeding, if you fed them versus starve them, if this can change the physiological responses to the Vibrio?
This is something that we didn't test, but I think it could possibly add to, yeah, for sure.
Thanks.
Okay, Sarah.
Thank you very much, Anna. For me, it's a very complex subject, but you made it quite clear, so congratulations for this. I have a first question on your slide 16, where you're analyzing the change of the effect. Yeah, you say that you have no significant change on the NRF, but you see the same trend than in the paper of by Seneca et al. In 2020. Did they found a significant change in this paper, in this other paper? And in any case, why, in your opinion, the change is not significant?
So in this study, yes, they saw a significant change. This was also on the transcript level only. And here we saw the same trend because the study analyzed the infection only up until 12 hours. So this is what we're looking at. The trend is similar at the 12 hours because they don't know what exactly happens after that. Yes. Sorry, you have another.
The second part of the question was, in your opinion, why in your case it's not significant? Do you misreplicate? What is your...
I think it's just due to the fact that there is a lot of variability in my experiment, and also the concentration of BP used in the two experiments is different. In the paper, they used 10 to the power of 8 CFU, but here I exposed them only to 6 times 10 to the power of 3. So I feel like that has definitely a significant effect on the gene expression levels.
Thank you. And can you just go further where you show the effect also on the epcase 3 and 8? Yeah, in this graph. So my question is, could you explain in your own word, in an easy way, so what is this process of cascade from the NLR to the epcase 3 and 8? What does it mean? If you should explain it to people that are not in the field.
So this is only a hypothesis that we have, so I'm not sure if it's actually happening this way. But a very common thing that has been observed is that, first of all, in what we know about in humans is that caspase 8 directly activates caspase 3. And NLRs are known to interact a lot with caspases. For example, like I talked about in the inflammasome complexes, the NLRs bind with caspases and activate caspases, for example. But I don't know if I answered your question correctly, though.
Partly. So could you just explain what are the role of epcase 3, epcase 8 for someone that is really not in your field?
Okay.
For common people.
Caspases are enzymes, and they usually cleave things, so they cut and activate stuff. So, yeah, for example, what we know about caspase 3 in humans is that it directly cleaves gasdermin, which leads to the activation of pyroptosis.
What about the 8?
In humans, again, what we know, because in ataxia it's still not studied, so we can't be sure. But in humans, we know for sure that caspase 8 is activating other caspases in a cascade, so that they all work together, basically.
Thank you, Anna.
Thank you, Sarah. So we can go back with the members of the jury online. Let's start with Stefano and goes on with Eric.
Okay, Anna, thank you very much for the... Can you hear me?
Yes.
Okay, so thank you very much for the clear presentation and the clear thesis report. Was very nice to read it. Really nice piece of work and discussion. Yeah, I enjoy. I learn a lot of stuff. It's not my domain, so I have plenty of curiosity, but I will reduce to just a couple of questions, I suppose. Well, first of all, there is I really like it. There is one thing missing that's more my domain. It's not missing, it's my curiosity. About you show a tree in your presentation that I would have liked to see in your report. What can you tell me about the distribution of these molecules that seems so different? Well, they're one from the other. I mean, it seems very ancestral, and they're missing in some of the canonical animals. So can you tell me something about the phylogeny and the evolution, what you know about these molecules?
Yeah, so with NLRs, what we have seen is that the fact that cnidarians have them is a very important thing, because cnidarians are really early diverging metazoans. Therefore, the common ancestor between Bilateria and Cnidaria both had inflammation potential, as well as apoptosis has been linked with this level. Through evolution, we see that the domain structures have changed. For example, in plants, the domains are very different, as you can also see in the as you can see in the picture here. And yes, in fact, it is true that some of them have fully lost NLRs, but a lot of animals have them. They are very important components.
Okay, so the 5 and the 3, are they orthologous of which one in vertebrates, for example, in humans? So they are paralogous, they are orthologous. What is the parenthood between the human molecules and the...
So NLRC3 was named NLRC3 due to the fact that it is homologous to the human NLRC3. It has some differences in domain structure, but they are very similar. Sorry, I don't know how to do this here. No.
Yeah, there is a transmembrane that is not supposed to
exactly.
have in the vertebrates, right?
Yes, but also at the same time, if you look at the similarity with the human ones, they are highly similar. And the NLRC5 is also the homologue of human NLRC5.
Okay. And since you are in this this slide, so what you are showing here is the results of the analysis of the transcriptomes, that
Yes.
transcriptomic analysis. Okay, so you already see that you have the caspase 3 and 8, that it seems to be upregulated before the upregulation of the NLRC5. So that shouldn't that give you the idea that the apoptosis is in this case is triggered before the expression of the NLR molecules? So what would you expect that the NLR is upstream the apoptotic event? Because this suggests quite, unless I misunderstood the data, this suggests quite the opposite.
Yes, this is something that I haven't considered before, but in fact, it might be upregulated before. But also, we are not sure if these exactly are the two NLRs which are the ones activating the caspases, because there are many more NLR sequences in Aptasia.
Okay,
these are just the two.
Because that's a little bit what you see in okay, they are not statistically significant, the NLRC5 and 3 your qPCR, but it suggests a little bit, if I look at page 25, if I look at the expression of upregulation of the caspase and then the upregulation of the NLRC5 and NLRC3, it kind of mimic what you are seeing in your transcriptomic data set. So again, we are just talking because there is not statistical support, but I don't know, think about that, because they may be in some way unrelated. So they're so diverse and pleiotropic, these genes. So
I think also it's important to remember that caspases get activated by many things.
Mm-hmm.
So it's not only
NLR.
Yeah.
Absolutely. Do you have time for another question?
You do.
So
fair enough.
Yeah, so it's something a little bit, it's a curiosity more technical. So for your qPCR, usually, I mean, I remember we use the delta CT approach for generally, but it's not exactly what you use. And why is there a reason for choosing this different approach in your analysis?
Well, actually, it is very close to that approach. It's just using different nomenclature. I can show you the formulas to calculate all the values. So it's the delta CT is basically the relative quantification, the RQ.
Okay. Yeah. My bad. I overlooked that part. So,
yeah, no worries.
Yeah, and for to optimize another technical question and curiosity. So to optimize the transfections, that it didn't seem to work. What do you have in mind? Did you think with your colleagues of something to optimize your cell transfections, or? I have no idea what's the cell biology on the Cnidarian cell. To transfect Cnidarian's genes. How?
So using the transfections, they discovered that there were some problems with the plasmids. So actually transfecting the plasmids was possible in the human cell line very well of the aptasia sequences. It's just that there were some problems with the plasmids and therefore it didn't work out the right way. But in fact, it was done perfectly by one of my supervisors just a couple of weeks after mine failed, so to say. Yeah.
Okay.
So it is possible.
Okay, so problem solved, basically.
Yes, exactly.
Okay. Fine. Well, again, I mean, they have to stop me because I have other questions. But something for Eric.
Can I invite you, Stefano? Maybe I can invite you, Stefano, to give the floor to Eric, and we may have time to come back to your last question. Eric?
Thanks, Anna.
Sure. I start with what Stefano just said. Thanks a lot, Anna. I had much pleasure reading your manuscript and also listening to your presentation today. Very, very clear, very focused. Much appreciated. It's a little bit tricky to come with my list of questions that I progressively strike through because most of the questions I had, they came already. So I will come back to some of the aspects that were already asked. It's sort of follow-up questions. You mentioned that the NLRs are extremely important in all animals. However, they're missing in Drosophila and C. elegans. A lot of genes are missing in those critters. They live in rotten fruits, so I think they're not in a very sterile environment. Do you know how the innate immune system works without the NLRs?
So there are other very important receptors called toll-like receptors, and these are present in Drosophila and C. elegans, for example. So I think that they have a very big role in innate immune responses. But other than that, there are many other receptors. So it's not only NLRs making sure that the innate immune response functions.
Okay, I'm not sure, but I think the toll-like receptor is also present in Cnidarians, so it's a very vast family. Okay. Here, you showed the phenotypes at the different concentrations, and you showed at the end the most drastic phenotype is liquefaction of those poor sea anemones. In those previous concentrations, they look quite compacted. They lost the tentacles, but they still looked, for me, looking at the panels, quite alive. So my question here is, if you put them back into non-infected media and rinse the water regularly, can you rescue them?
Yes, in fact, all the anemones that survived from this experiment, we put them in fresh water and we fed them, we put them in light again. We kept changing the water as we maintained the anemones every day, and they were fine and they were recovering well.
And then question, if you reinfect those, are they more sensitive to the same concentrations or more resistant?
So sadly, this is a step that I didn't carry out yet, but would be interesting to look at in the future.
Okay, yeah, we'll be curious. Would be interesting to know. Then I have a sort of a conceptual question. It's not my field, but for me, sort of genes involved in the immune system should try to avoid to kill the cells or the organism. Here, part of your study was you were looking at the role of the NLRs activating at the transcriptional level, even if you said that they form complexes at the protein level, which is a different layer. So my question is that I don't really get the point of a immune system gene that tries to fight against stressors by killing the cells that are infected. Can you sort of comment a little bit, please?
So apoptosis is a very important defense mechanism for all multicellular organisms. And it is really necessary for getting rid of the cells that have become toxic to the organism. I'm sorry, could you repeat your question one more time?
No, it's. I think I don't have the answer for this, but my question is really I try to understand why the immune system would foster apoptosis, because that's the end point. That's I think all that you would like to avoid is killing the cells or the organisms. Okay, so that's the last step to do. So why are the NLR doing this? It comes up probably only at late stages. And so my question was really more conceptual, is trying to understand what the connection is between the innate immune system and trying to push towards apoptosis.
So apoptosis was shown to be a very important mechanism, like I said, especially on all multicellular organisms. And it really is something that controls the way that they first, it has been shown to be also at the very early diverging animals. So it must have been a mechanism that really shaped how multicellular organisms became to be so functional, and how to better control where the cells proliferate, where they don't. And so I think on a functional level, it is one of the most important mechanisms evolutionarily for us even. I'm not sure if I still answered. I'm sorry.
No, I won't push you too much into this, because as I said, it's a conceptual question. It's not a yes, no or something.
Okay.
It's here the idea is that you have all the cells that get sort of the same load of Vibrio. Okay, so the question behind my question was: do you think that there's a specific cell types that undergoes apoptosis to allow the other ones to survive? And as Aptasia, like most Cnidarians, are extremely regenerative, they simply sort of replace the dead cells. And so
Yeah, for sure.
that was sort of why I tried to connect the dots.
Yes, definitely. And in fact, with the pyroptosis pathways, the way that the cell forms pores in the membrane to release pro-inflammatory cytokines, letting the other cells know what's happening. And so I think that that is a very essential step in exactly controlling which cells die, which don't, how to better control the defense of the organism now.
And the final question: do you have any evidence by looking
Really
at
last question? Sorry, Eric.
animals that survived, if there are? Sorry, last question.
Yes, a short last question with a short answer. Sorry.
Okay, just do you have any signs of specific cells that die after infection by looking at the animals?
Through visual observation, I can't distinguish which ones are dying, but as you can see, the end result, all of them die in the end.
Okay.
Yes.
Thanks a lot again, Anna.
Thank you. Congratulations, Anna. Before you leave, you can see some comments on the screen in front of you from Cass, who is clearly not working, Emma, and Melissa, and Rita.
Thank you.
Right again, so we can go to the next presentation. We're going to start in a second.
All right, so if it's okay, I'm going to get started. So good afternoon, everyone, and thank you for being here today as we present our final master's thesis, culminating our 2-year journey with MARRES. My name is Callie Paul, and today I'm going to talk to you about the transcriptomic analysis that I conducted for this. We have crinoidea, ophiordia, holothordia, and astroidea. Now, in this particular master's thesis study, we are going to be focusing on the subclass of Echinoidea, which consists of sea urchins. Now, echinoderms are very unique as they most exhibit a biphasic lifestyle. So in my particular study, we're focusing on the sea urchin Paracentrotus lividus, which has been characterized and used as a developmental model in biology for over a century due to its access of easy development within a laboratory setting and its biphasic lifestyle. So what I mean by a biphasic lifestyle is that in early development, we are at a planktrophic stage where the gluteus is expressed in a bilateral symmetry, and we meaning the echinoderms, undergo a dramatic metamorphosis, which after their body plan changes to a radial or pentaradial symmetry. Now, some specific attributes of the Paracentrotus lividus echinoderm is that there's phenotypic variability, meaning that the color can be expressed in purple, green, brown, etc. While the habitat distribution of P. lividus extends globally, this species is highly present in the Mediterranean, and actually their edible gonads are considered a culinary delicacy. An interesting structure in echinoderms is actually Aristotle's lantern. It's historically relevant as well, as Aristotle characterized this mechanism himself, and it is used as a masticatory structure in echinoderms. As I mentioned earlier, P. lividus exhibits a biphasic lifestyle, and in the adult period, it displays radial symmetry in pentaradial form. Now, here, this symmetry is displayed as we have 5 umbilica and 5 interumbilica that ultimately comprise the urchin test, also known as the shell. The water vascular system is used for motility. And to give you a general overview, water enters through the madrepore, filtering throughout the various canals to ultimately inflate the tube feet poda, allowing the sea urchins to move in their adult stage. So while we are understanding that these are some characteristics of echinoderms, specifically P. lividus as well, a former member of my team, Laurent Formery et al., actually at my current team, Evo Inside, highly developed a characteristic of the P. lividus life cycle, denoting 3 distinct periods comprising of the entire life cycle. So it's important to note the colors, as these will be uniform throughout the rest of my presentation. We have the embryonic period in yellow, the larval period in green, and the adult period in blue. Now, what you're seeing on the right is not meant to be read, but it's just to characterize how detailed this life cycle assessment was. And each row is designated into 33 further characterized developmental stages within the developmental periods, the embryonic, larval, and adult stages. Now, it's important to note that within the larval period, green, we have 15 additional stages that characterize rudiment development. Rudiment is very important in echinoderms as, it comprises the adult body plan. So this small structure here in the bilateral larval stage actually engulfs the entire larval body, much like when you take off a glove. And the resulting body plan is a pentaradial form beginning after metamorphosis in the first period of the adult period, the early juvenile developmental stage. Now, as you can see, the P. lividus life cycle has been very well characterized based on morphological observations using microscopes and also in situ hybridization. Now, what hasn't been fully elucidated is the molecular mechanisms that drive such dramatic morphological variations across development. And this is where my master's thesis comes into play, addressing the question of which genes are involved in coordinating major morphological transitions across Paracentrotus lividus development. Now, within my study, I utilized an RNA-seq transcriptomic analysis to address this question. To give you a very brief overview, prior to my arrival in Villefranche, various extractions of mRNA and biological replicates were taken temporally at different stages throughout development. Those mRNA extractions were then sent to BGI Hong Kong to essentially be sequenced through library generation and next generation sequencing. Now, when the raw data was sent back to my lab, this is where I came into play. I utilized this workflow using a combination of Python and RNA lysis to essentially transform these raw reads into transcripts per million and estimated counts. Transcript per million gives you an idea of how many transcripts are available within the dataset at one given time. And the estimated counts give you an idea of the number of genes that are witnessed throughout the transcriptome. Now, once I had it in tangible units, I then utilized RStudio and DSEC2 to get a better idea of how these genes are behaving across development and any similarities. So in order to further validate the homogeneity of the mRNA extractions that occurred prior to my arrival, my first step in this analysis was to take 13 of these stages, represented once again, yellow embryonic, green larval, dark green larval rudiment, and blue adult stages. Taking these 13 stages, I then conducted a principal component analysis, which ultimately represents the 13 stages that I mentioned previously, denoted by different colors, once again denoted in the different developmental periods. And each of the biological replicates are marked with a triangle or a circle. Now, what we can see from this PCA is the homogeneity of the replicates, as we have very little variation between. What's also very interesting about this principal component analysis is, if you notice, the chronological occurrence of variation. What I mean by this is that our first embryonic stage, we have the swimming blastula, which is the first stage of the embryonic period in this study, followed by late mesenchyme blastula, and so on. So what we can say is the embryonic period is represented here, following through the larval period, larval rudiment development, and the first stage of the adult period at the early juvenile developmental stage. Now, once I confirmed the homogeneity of the replicates, I concluded that this data was efficient and I could further explore how these genes are behaving across the 13 stages. Now, in order to do so, I conducted a differential expression analysis using the 13 stages. And what we can see along the x-axis is, once again, the developmental stages utilized in accordance with their developmental period. And on the y, we see the number of genes. So what this shows is that the blue represents the number of upregulated genes between one developmental stage to the next, and the orange denotes the number of downregulated genes from one developmental stage to the next. The main takeaway from this differential expression analysis is that we see a large number of upregulated genes within the first 6 comparisons. And at the last developmental comparison, from the larval rudiment stage to early juvenile of the adult period, we see an increase of downregulation of genes. Now, in order to conduct a more focused analysis, I selected 6 developmental stages, or 3 comparisons, to further focus the analysis. It's important to note that each of these developmental stages also incorporate each of the embryonic, larval rudiment, and early adult stages or embryonic periods. So we have a full view while also keeping the trends that we saw in the original 12-stage comparison. 13-stage, apology. Now, from here, I conducted a completely different differential expression analysis. And as you can see, the trends observed are very similar. However, the normalization changed as I went from normalizing 13 developmental stages to 6. So what this did was actually remove false positives. And I know you might not notice it right away, but in the last comparison, we actually saw an increase of the number of downregulated genes observed throughout these comparisons. Now, we denote that between the 12 comparisons and the 3 comparisons, the trends are similar, with upregulation in the beginning, first comparisons, and an increase in downregulations at the last comparative stage. In order to further explore how these genes are linked, what these genes could possibly do, I conducted a hierarchical clustering analysis, which elucidated that these genes within those 6 developmental stages are best represented by 7 clusters. What you're looking at here is genes grouped by similarity. So how their expression levels are, if they're similar, they're grouped. It's also important to know that each one line represents an average. There are hundreds of genes within each cluster, and for time's sake, I'm just going to elaborate on cluster 6, which is shown in black and expresses upregulation at each comparison. Now, in order to get an individual view of each gene and how it's expressed across, I then conducted an individual cluster analysis. There are 148 genes in cluster 6, and each line denotes the expression levels of a gene. The overall trend exhibited in cluster 6 is upregulation between each of the stages, and this is a normalized TPM. Now, in order to actually get a grasp or an idea of the functionality of this cluster, because right now we see the patterns of the genes, but we don't know what these genes are, I then conducted a gene ontology analysis. And so what I did is I took the 148 genes of Paracentrotus lividus and I blasted them against sister species Strugulentrotus preparatus, which is another sea urchin species that has well-annotated genes. What the BLAST analysis showed, in accordance with ShinyGo, which is a gene ontology tool, was that cluster 6 is most enriched for energy and metabolic processes. These processes are then subdivided into groups, and within those groups, they were further categorized into 28 categories, subdividing the enrichment groups. From those 28 categories, I then selected the category biosynthetic processes as it had the highest number of genes, and noted that there was NOS, or nitric oxide synthase, present in this cluster. I then took that gene, went back to the transcriptomic analysis that I previously conducted, and elaborated on the expression across the 6 stages of NOS, or nitric oxide synthase. What's interesting about NOS is that we see a similar pattern of up expression of genes, or highly expressed in the first 4 developmental stages, with almost no expression observed at the early juvenile or first adult period. Now, this follows the trend of the differential expression analysis and also further elucidates as to what the possible molecular mechanisms that are attributing to the morphological variations across Paracentrotus lividus. This also highlights what the possible function of nitric oxide synthase is in the neural mechanisms across morphological transitions or development. I also conducted a literary analysis which elucidated the fact that in 2007 and in 2003, Bishop and Brandhurst spatially confirmed the expression of nitric oxide synthase, and it was shown to be in the ciliary bands, and this is on a 4-arm Pluteus of a similar, an echinoderm of L. pictus, which is another sea urchin. Now, ultimately, the RNA-seq transcriptomic analysis is a very robust way to look at how genes are behaving across a developmental period in time and can give further elucidations as to how these molecular mechanisms work to assist changes in morphology or create developments such through metamorphosis and so on. Ultimately, the PCA confirmed the homogeneity of the samples and also exhibited chronological variance that was in parallel with the developmental stages characterized by Formery et al. The differential expression analysis showed that an upregulation trend was observed from comparisons 1 through 6, and a downregulation trend was observed from the last larval rudiment stage to the first adult period of the early juvenile developmental stage. Gene enrichment analysis concurred that energy and metabolic processes was best enriched for cluster 6. Something that I conducted in my master's thesis but did not include in this presentation was the validation of suitable techniques to further confirm the Paracentrotus lividus transcriptomes. And I concurred that chromogenic in situ hybridization was best for embryos, and fluorescent in situ hybridization was best for larval stages of Paracentrotus lividus. Future perspectives include further spatial expression to confirm the gene profile results that I obtained in this transcriptomic analysis, and to ultimately discern where and when the genes are expressed across development. An expansion of the cluster exploration could also further elucidate biological functions of each cluster. And this master's thesis also provides a basis to explore more focused analysis, such as neural gene conservation across P. lividus, as these are just some examples of the neural genes that I defined and denoted their profiles in this master's study. I just want to take a moment to thank my host laboratory and my team, Evo Inside. I would like to thank Anna, Yanni, and Lotta for being with me at every step of the way through this 2-year process. I would also like to thank Dr. Adrienne Poquet for existing at the same time as me, for Pascal, for Az, for Marion, to Tilu and Mateo for providing energy in the laboratory every day. A big, big thank you, and a very special thank you to Dr. Joelle Caveril for answering any and all of my questions, no matter how naive. Thank you.
Thank you, Kelly. So we are now going to questions. As we finished last time with Stefano and Eric, I propose Eric to start the questions and Stefano to take over. Eric?
That's good. Thanks, Christoph. Thanks, Kali, for the nice and clear presentation. I also liked the introduction, the first part of your manuscript, and I got a little bit lost in the result section. And one of the reasons I think, and I have the same question here, is that I perhaps didn't get the reason why you started to do a differential gene expression analysis without doing the annotation of the transcriptome first, because you have lots of genes, or perhaps I missed it, and I missed it again. And here you even don't really talk about it, that this should be done in your analysis. In the manuscript, you talk about it. So for me, you jumped right into differential gene expression analysis of comparing things without mentioning why you focused on comparing the different stages. I understand the whole process, but not the individual comparisons. Or can you clarify this a little bit, please?
Yeah, thank you. And so there's multiple answers. So first, I just want to say that the P. lividus transcriptome, we have a limitation on the annotation. And much of the annotation that I obtained was through BLAST. So it was comparing the sequences or the gene profiles to Strongylotrotus purpuratus, which has been well characterized. But ultimately, what's something very important about this analysis, so I'm glad you asked, is that in this analysis, we started from a very broad spectrum and really followed the data to follow the patterns first before selecting candidate genes. And a lot of papers that I read, they do the opposite approach, where they select a certain number of candidate genes. Let's say they have a lab that focused on neurogenesis, much like my lab. And we actually had the idea of, okay, should we take some neural candidate genes and then specifically identify their profiles throughout development? Or should we take the holistic approach, follow what the data shows based on gene expression levels, and kind of siphon through to a more narrowed approach based on what we saw on what was being expressed and how and when. So that's the approach that we took to attempt to exclude as much bias as possible. But yeah, that's basically my understanding of it. We kind of went the opposite way and then selected the genes at the end based on what we saw on the gene ontology and what we saw on the cluster profiles. While a lot of other studies that I read, they select around 10 genes, they annotate the 10 genes, and then they work in reverse to ultimately discern how they're expressed and when, and then some even use functional analysis to determine the direct link on how they're involved in development of certain species.
I totally agree. I think the whole goal of doing a transcriptomic approach is to have an unbiased approach, and you clearly had it. It's less about the approach. It's more about if you have the transcriptomic information or the annotation of the transcriptome, then you can easily go and see what's going on, and you don't have to wait until the end of the analysis to do what you did. I understand if there's a technical issues, you can't do it at the beginning and then you do it later.
Right.
So that's fair enough answer. But I think it's clear if you do a transcriptomic analysis, that's to stay unbiased and then try to see what the biology tells us. And I still, you want to add something? Sorry.
No, just thanks.
No. And I still come back to the comparisons, the different gene expression you did at the beginning, and then you trimmed it down to 3 or 6.
Yes.
Why did you choose those?
So these 6 developmental stages, they incorporate the prism and the larval. So I already stated this. Essentially, it follows the trends of what we see in the overall expression, but it's also based on the characterizations that have been previously defined by Laurent Formery. So we know at the prism stage, that's the last stage in gastrulation before we move into the larval. And so ultimately, we have dramatic changes occurring across the development of Paracentrotus lividus. I know, as you studied. But the idea was to ultimately determine how are the genes behaving at the embryonic stage compared to the larval stage. How are the genes then behaving from the larval to the larval rudiment? Because at the larval rudiment developmental stage, we have the formation of the larval rudiment, which ultimately determines the body plan. So we have a lot of significant morphological changes occurring, and this perspective then includes each of the developmental periods while also keeping the overall trends. And so ultimately, in short, based on the morphological observations that we made previously and what we observed in the trends, we chose 6 to focus and also to narrow the normalization to be sure that we weren't including false positives in the results, and to narrow the global analysis to select a further number of genes. It's also important to note the vast amount of genes that are actually involved in this study. Ultimately, in this differential expression analysis, there's approximately 11,000 genes that we're working with. So in order to filter those genes, these were the measures that we took to try to go ahead at ultimately lowering the number of genes in this analysis to potentially select candidate genes without biasly selecting neural genes or biasly selecting genes with a specific function, if that's clear, sorry.
Right. Well, it's clear, but I'm still a little bit missing the answer to my question. Why did you choose those 3 transitions? Because you have 13 to choose from.
Yes, because they include the overall trend while also including each of the developmental periods from embryonic to larval, larval to rudiment, and larval rudiment to early adult.
Okay, I missed that then. Okay.
Sorry.
My last question, then I give it over to Stefano. You didn't present it here, or very briefly at the end. You did the in situs and the fluorescent in situs, HDR, and you conclude that one is good for embryonic development and the other one is good for late stages. But you didn't do or at least I missed it, the in situ with the fluorescent probes on the embryonic stages, did you? Or why is it not working?
Yeah, actually we did. So ultimately there's many reasons, but to give you the very brief answer, and then I'll elaborate a bit more, is that ultimately the larval body, when it's subjected to the heat, because the temperatures of both of the methodologies, for example, the whole mount in situ, the chromogenic, is at a higher heat. It's about 67 degrees that they're fixed at, and for the fluorescent in situ, it's about 37 degrees. So if you actually look at what we observed at the late gastrula, we have the body, it's fixed, the embryo appears to be intact, and we're seeing the expression of FOXQ2 at the apical origin. But when we move to the 2-arm, we lose the morphology. So I can't denote where this gene is being expressed at this stage because I can't denote the morphological structure or align it to what we know. So if I compare then to the fluorescent in situ hybridization, the A is going to be the mesenchyme blastula, which is also going to be at the embryonic stage. And while it's well represented, in this specific case, this is synaptotegmin B expression. It's not known to be expressed at these stages based on the literary. But if we go and compare this 2-arm Pluteus stage, we can see that the morphology is distinct, so we can identify where the genes are actually being expressed. I don't have a complete concrete answer, but when discussing with my team, the most obvious thing to me was the differences in temperature that are adapted by both methodologies, and mostly that the fluorescent in situ hybridization is highly utilized within my team to ultimately denote the expression of the genes and where they're being expressed.
Right, I get that. So I totally agree that for the late stages, you don't affect the morphology, and that might be clearly linked to the temperature. And I remember when I worked on left-right asymmetries in Syrian, it was a nightmare.
Yeah.
And my question here was really focusing on the early stages, because here you show a gene that is not expressed during embryonic development or mesenchyme blastula stages. And so you can't conclude in saying that the fluorescent approach is not usable during those stages. It's more about that, because you say that you use one for early stages and one for later stages. But if you can use one for all the stages.
Yeah, it's more, yeah, for sure. And ultimately, it's not to say that fluorescent in situ hybridization cannot be used. It's more suitable for specific developmental periods.
Okay. All good. Thanks a lot, Kali.
Thank you, Eric. Here we go for Stefano. Stefano.
Yes, here I am. Thanks, Kali, for the presentation and for sharing your manuscript. So I'm kind of I kind of shared the same. Well, I was the same idea that Eric has. I was surprised that I didn't see an annotation, but I don't. Let's not go through that anymore. I was also looking for, when I started to read your manuscript, for some more geeky and juicy details of the analysis in the material methods that unfortunately I didn't find it. So, and it wasn't clear to me. So, from which step did you take over this data set? Because you have a very nice data set to play with. And you started from what? From the raw reads?
From the raw reads.
Okay, so if you start from the raw reads, I would have liked to see also the boring stuff. I mean, how were these reads? How are the quality? And here we are. There are the after slides.
For sure.
And,
so we did. I used RNA lysis, the Callisto paired end reads. So essentially, it takes the read, it cuts it, and you have from the opposite end, it reads it this way. So it's a way to further validate the results that you get from the sequence. Sorry, this is not the slide I want to be on.
yeah.
Here we go. So yeah, and from there, actually, this process is quite lengthy because you have a lot of data. So essentially, we use pseudo-alignment. Pseudo-alignment is differing from the normal alignment reads, as instead of taking every single nucleotide that's there for every single gene sequence, it takes a reference point, and from that reference point aligns to the nucleotides that are in question or most sequential. So it's a faster way to do this analysis because ultimately just transforming the data from the raw reads into the TPMs or estimated counts was about a month of work. So it takes more time, but using the pseudo alignment, it is a more rapid process. But that's the format that we got them in, and they're 150 nucleotides long for the paired end.
Okay, so if I understood, you align on the genome, right? So you map on the genome your.
Yes, but it's a pseudo alignment, so it's not directly. It's just the nucleotides in question based on the index that's generated from the Callisto RNA lysis. Okay. So, and you choose this straight approach just for a question of time, of saving time. Exactly. The pseudo alignment is more rapid, and previous studies actually take each methodology of sequencing and aligning these methods, and based on time, they determine whether the quality or not is efficient. And pseudo alignment has been compared to regular alignment as just as efficient, producing well-valued results as well. Okay, so yeah, a little bit more details on that on the material and method. I would have, because you use Callisto, right? For the Callisto paired end quantification. Okay, so yeah.
Stefano, maybe a last question.
Yeah, sure. So the last question is, well, I'm happy that you showed, you talk about one gene, at least in your presentation, that is not in you talk about NOS, right? Yes. So what does it do and why you choose that among the others? So this was where the bias came in. So I essentially selected nitric oxide synthase based on the literary analysis. It was also proven, or not proven, but elucidated that the downregulation of nitric oxide synthase in 2003, also by Bishop and Brandhurst, may be in accordance with the signaling for metamorphosis. But ultimately, it's been defined in the ciliary bands and has been noted that it defines neurons, or it's a neural signaling molecule. To be clear, I finished the clustering results after I submitted the thesis, so I know that you're missing a part of this. So if I could just show you very rapidly. Doesn't matter. I did the cluster analysis for each of the 7 clusters. For example, in cluster 3, it was most enriched for cilia and motility. And I actually went back and selected RIB74, which has been defined to actually produce the flagellum. And the expression profiles, once again, follow the trend expressed in the differential expression analysis. But ultimately, it's 100% correct.
Further insights into the clustering analysis needs to be elucidated to identify specific target genes, to then relate them to how the gene expression behaves over development, to have more insights as to which genes are directly related to processes.
Good. Thanks, Callie.
Thank you.
Thank you, Stefano. So we are back here with you, Sarah.
Thank you, Callie. It was a very nice and pleasant presentation.
Thank you.
I have a question about your slide 7 with the principal component analysis.
Yes.
Usually we do this analysis to summarize the sample and to find a cluster of samples. So can you just explain us which are the information, the input information, the variables that you put in your analysis?
These are estimated counts of the expression.
Only the estimated count?
Yes, so I did many PCAs actually just to visualize what the data was doing, but ultimately to confirm the homogeneity of the mRNA replicates, because we had 2 replicates, and we wanted to see what the variance was across development as a starting point to then guide us to the next analysis. But this is an estimated counts.
Okay, and usually we use this analysis to interpret which pattern we see in the to the axis. So if you should you explain us a bit the x-axis, but what about the vertical axis? What is your interpretation from?
So essentially, this accounts for 60% of the variance this way. So ultimately, this entire PCA accounts for 85% of the variance included. To simplify, each square represents around 7.5% of variance. So what you can determine from this principal component analysis is you can presumptuously group the stages by proximity. So as I stated, these designate the embryonic period, then moving through the larval period based on proximity. I didn't actually conduct vectors because you can actually mathematically determine the exact variance between the points. But as I said, roughly 7.5% variance is accounted for. And then ultimately you could make groups based on how they're clustered. And what we observed that was very interesting is that the variance occurs chronologically in parallel with the characterized development by Formery et al.
Thank you. And just a quick general question. So I understand that this kind of analysis is the first time that were carried on sea urchin. But do you have other example of literature where they process the same analysis to understand which are the molecular driver of the morphological change?
Yeah, exactly. And this wasn't the first RNA-seq on echinoderms, sorry. This is P. lividus, actually, from the Villefranche laboratory. They just produced a paper specifically on Paracentrovis lividus on an RNA-seq, and they actually did an attack-seq, so it's a little bit different. But RNA-seq is an extremely robust tool because you can essentially visualize how the genes are behaving across development. So in other species, as you asked, this has been done in Astroidia. A lot of the papers that I referenced have additional transcriptomic analysis conducted. And as Eric was touching, normally these analyses have a specific path in mind. So let's say you want to keep talking about neurons because my lab works in neurogenesis. But for example, there is a paper that talks about the expression of genes across development of Astroidia, and they selected 10 candidate genes. So while they did the differential expression analysis, just to observe the overall trends, ultimately a biased choice was made to select the 10 genes of their interest for their particular lab. And then it does provide, like Stefano said, more insights as to the actual function and how those genes are related to the morphological transitions. But this is a super robust tool, and there's many different ways that you can go with the analysis, and it's a very broad way of analyzing development.
Thank you, Kelly, for this amazing presentation. So it's great that you have this idea. I have one question regarding that at biological level. So I can get that the beginning of your curves, so all the different stages are really quite different.
Right.
Then they are quite similar.
Right.
So down all the, all the cell differentiation and so on may have a few already for some cell. But I don't get why the early juvenile slightly come back.
Right, for the variants.
At the biological level.
At a biological level, I found this interesting as well, but I'm going to answer you, but I'm going to go back just a little bit. What's very drastic about echinoderms in general is post-metamorphosis, you have a completely different body plan and structure. It's almost alien-like. And what's very interesting and what a big question is how conserved are these genes across? And what we noticed based on, if I take one gene, we have these upregulation and post-metamorphosis, we have an increase of downregulation or almost no expression of the genes. So for me, on a biological level and a very naive level, I also pair this with what was characterized in the life cycle studies defining morphological differences. And while the variance comes back, these can be grouped a little bit, though the variance is still accounted for as the vector. So ultimately, for me, the reason that the E. juvenile is different is because of the different morphology that's undergone and the different expression of genes. As this is a very naive statement, as I didn't actually look into specific candidate genes, we noticed a completely different expression from the first embryonic and larval periods to the adult periods, indicating maybe there are other gene cohorts at work that are then turned on or expressed but differ from the adult. I just want to note, though, that the early juvenile is actually not the most variant developmental stage. And if you look, it's actually going to be at like the forum pluteus. So that was interesting as you have a transition from the embryonic period where you're one embryo and you move to the forum where actually feeding starts and you have starting arm development. But I actually didn't expect this. I thought that based on this data, the adult period and the early juvenile would be the most variant of all of the stages based on what we know about the morphology.
And one last question. So you start right away. So often what we use is also having a gen approach. And you know any way to interfere with the metamorphosis in such system? And do you have any idea about which kind of pathway you can target to also perform an RNA-seq, but in a
functional way, for sure. So that's what's super interesting about this project is it can be used as a baseline to explore functional analysis, which is not something that I was able to touch. But for example, for nitric oxide synthase, as we know that we indicate based on previous studies that the downregulation may incite metamorphosis, possibly doing a knockout or a knockdown of the function. You could then essentially take a control where you have we have it. So, sorry. You could take this profile, and if you wanted to target specifically this gene that's associated with metamorphosis, and then you could do different functional expression analysis to inhibit the function of this gene. And then you would go back and extract the mRNA after the functional analysis to then compare how it's expressed. But you could also do that visually, observing the morphology across development of different samples.
Okay, thank you.
Thank you.
Okay, thank you. That's a deal. So we have arrived to the end of your presentation. And so I would like, I think, just before you congratulate again, have a look to your fan club as usual. So first, Britta has something to tell you. It's written on the screen.
Thank you, Britta.
The dream in pink, saying that. And we have then Britta again, Marta, Kilian, Erwin, Melissa, Doug, Emma, Ina, I'm going a little fast, sorry, Shani, Paul.
My sister.
And Dominion.
Chloe. Hi, Chloe.
Paul. Okay, so congratulations again.
Thanks again to the jury for being here. And thanks for being here. And that's my notebook.
So we welcome back Camille. Hello Camille. Ça fait changer les écrans.
Can you hear me now?
We hear you, yes.
Great.
So we just need a second just to change the screen, which, as you can see, is in front of us. So, Emily, do you have a countdown timer in front of you?
I do.
Good. So you're currently in Woodsole. As I like to ask to all the students online, what time is it in Woodsole?
Right now it's 9:26.
Okay, good morning. Good morning. Yes, that's a good morning, Camille. If you're ready, I can offer you to start. You have the floor for the next 15 minutes.
Okay. I'm gonna put my presentation. Everything is good? You see everything?
Yes, we see an octopus.
Perfect. So to introduce my presentation, I would like to show you how a natural octopus blindly forages. And so as you can see here, this octopus is introducing arms in really narrow places. And this remarkable behavior is of the interest of many communities, from behavioral ecologists, neurologists, soft robotic engineer. And for that reason, I had the chance to develop my master thesis within Roger Hanlon laboratory, looking at octopus arm use and recruitment during blind manipulation of objects. When an octopus forages, what do they need? So first they need fine sensory abilities, and they need an accurate motor control of the arm. Looking at the sensory systems, so we distinguish 4 main sensory systems: vision, mechanotactile sensing, distance chemosensing, and contact chemoreception. What is interesting is that lately it has been demonstrated also that octopus are able to discriminate between prey, extract, and non-prey, as they will spend more time interacting with a prey object than a non-prey object. Also, when it comes to the arm use, it has been shown that there is what they call in literature a behavioral division of labor along the anterior and posterior body axis, where you will have anterior arm that will be used more for prey manipulation, while posterior arm will be used more for the crawling behavior. And when it comes to use multiple arm, you will often observe that when manipulating an object, often it's likely that the adjacent arm, so the anatomically closest arm, will be preferred to help in manipulating an object. But to do so, they need a fine coordination, and that's where the neuromuscular system comes. And a recent study also showed that you have neural connection with one arm and the first one over and the third one over, and that there will be potential connection between adjacent arm. And I insist on potential because it has been highlighted for some arm, but not for all of them. But the octopus arm choice is strongly influenced by eye use. And so that is why we wanted to ask, how does octopus, and specifically octopus bimaculoides, uses and recruit their arm when blindly searching for object? We based our hypothesis on what we observe in literature for the visual assays, so that anterior arm will still be preferred among posterior arm, but we also expected that we will to see specific arm per the different object that they will be exposed to. And we also expect that the principle of neighborliness, so neighborliness, the nearest neighbor involved when manipulating objects, will hold up. But as we are performing all of that blindly, we also observe to highlight potentially new other arm interaction. And so what we did, we developed a live animal behavioral assay where we exposed octopus bimaculoides to a set of several objects, and they were allowed to, let me put the pointer, they were allowed to blindly explore in a 3D printed dome, as you can see here. And so they had 3 weeks in total of experiment. The first week was an actual experiment where 9 octopus bimaculoides were tested, and they were exposed either on the morning and the afternoon, either to a prey or a non-prey object. On the first week, they were exposed to the non-prey object was a 3D rock shape. It was a 3D printed rock that was rubbed with shrimp so that they will be able to detect that distally the object. Then they had 1 break week. And then on the second week, only 8 octopuses were tested, and they were also exposed either to prey or to non-prey, which was this time a crab-infused agarose disc, so that they will be able to detect it not distally, because all the crab tests will be embedded in the agarose, but they will be able to detect it by contact. And I'll be happy to further answer why we had 9 octopus on the first week and 8 octopus on the second week. And so we basically recorded how the arm are behaving and how they are used and recruited using 3 angle. So a left view and a right view with GoPro camera and a front view with an iPhone. And so this is usually what would happen. So as you can see, there is a lot of thing going on here. You have some exploring arm, you have some contacting arm, you have what we call recruited arm, so the ones that are coming to help toward the achievement of retrieving the food out. And from all of this video, what we assessed, so we assessed 4 main components. The first arm to initiate contact, the overall arm use. Every time they were approaching the dome, we were looking at what the arm is doing in the dome. And the interaction and recruitment type, so when you have 1 contact, what is the second arm coming doing? And also the sequence, so all the interaction combined. When it comes to the interaction and recruitment type, as well as the sequence, we relabel the conventional name of the arm, as you can see here, by A being 1 arm contacting, and +1, +2, +3, +4, the anatomical position of the arm, depending on the arm that was contacting. And then to test whether there were general and specific differences in this different component, we use a set of statistical tests that I will be happy to elaborate further if you have questions. Regarding the results, here, what you can see on the left panel is like what were the most often used arm to initiate contact, and here is the most often used arm that are used overall in the whole assay. What is interesting that we could see is that the first time making contact was also the same one that was used most often in overall the assay. And also what I would like to point it out is that you could always see that it's L3 or L2, so left arm. And why so? It's because we think that the fact that we isolated our octopus on the left side before showing them the dome might have created a bias in the way they use their arm. And also, the most often used arm on the prey B assay is an anterior arm, while you could see that here on the prey A assay, so as a reminder, prey A is the first week that they were exposed to the prey B is the second week that they were exposed to the prey. And so prey A, you do see that it's posterior arm. So still, if you merge everything on the first arm making contact, you see that anterior arm were favored, but not posterior, not for prey A. And in the full assay, there were not any partitioning of anterior and posterior. They were used evenly for all assays. But now, when you look at non-prey assay, this pattern hold up with the 3D rock shape, where you still have the first arm making contact. The most often one used as the first contact is also used overall in the assay. But this do not happen when you looked at the crab-infused agarose disc assay, where here you have more anterior arm used, and here the one that is the most often used is a posterior arm, and it's left. And this, we explain it by the fact that for food trials, so the prey, they were able to retrieve the food out. And so overall, it created less variability between the first arm making contact and the overall assay, because once they retrieve the food out, they basically do not interact anymore with it, or we don't see the interaction with the front view. While for those ones, they will come, the data that you see here combine all the time that they approach the dome and all the time that they use their arm in this 10-minute assay. And because of that, it allows more variability. And so that is why you could see more disparities between the first arm and the overall assay. When you look, however, what is interesting is that when you look at the first time contacting, it seems that the first time contacting, anterior arm prevail over posterior arm used. And this is actually corroborating the previous result from Barn and al., which what is also interesting is that their trial include food. And for food, we did not have this preference of anterior arm. So that's why we were really intrigued on why. And we still really do not know the answer. And so we'll have to repeat to know. But for the full assay, we do not have an anterior arm preference for the crab-infused agarose disc, while you do have a clear one for 3D rock shape. And what we do think is that for 3D rock shape, although I will discuss further, they were not really interested in the shrimp extract, they still were able to detect distally the 3D rock shape. So they might be more interesting and go straight to the point. And so when they go straight to the point, we saw that they were using more their anterior arm, because that's the first one that come when they have to introduce it in the hole. Then when we looked at the interaction, so one arm coming and the other one interacting, what was interesting is that overall you have more direct recruitment. So direct recruitment is I make contact and then I have my other arm that is interacting simultaneously. So you have that for prey, but for non-prey assay, they tend to use a single arm to and not involving other arm. Although then after this single arm used, you'll have more indirect type of recruitment, which is contact for the non-prey assay. And also what was interesting is that there is a clear difference between prey A and prey B. So you still have more direct recruitment, but these nearest neighbors, so A+1 was consistently more used for prey A, while here you have other type of interaction, and actually A+2 seems to be used most often. And what we suggested is that you might have an adaptation on the way they use their arm with time. And so they might adapt the use of their arm between the first and the second week. So overall, the arm use and recruitment might have been influenced by several factors. So as always, you have inter-individual difference. So every individuals have their own behavioral specificities, or some of them were youngest. So our youngest in the cohort were 6 months, and they were white collected, which is another variable that might have influenced everything. So always this, the solution for that is to increase our sample size, which is currently ongoing. Also, when you looked at the contact, we evidenced on two types of contact, what we call a distal tap, which was usually a really short contact with the tip of the arm that was like usually less than a second. And another one that was median wrap, which involved more suckers. So suckers were really engaged on the object. And from that, we are not really sure what kind of information can be conveyed from such a short contact. So what we suggest is to conduct further experiment where we would put, and it's a project ongoing also, that where you will put electrode on specific region of the brain of the octopus to see whether you have specific signal that is transmitted when you have such short contact. And that will also allow us to see what is the degree of freedom from the central nervous system the arm have. Also, what was highlighted in Kawashima and Ikeda, as well as in Masali, is that you have a sensorial hierarchy where chemo, whether it's distance or contact, seems to play a way more important role in the overall other sensory system. And so that's something that should really be considered when you assess how octopus use their arm. But also what we saw, and it's also, I need to say, really dependent on your animal, because our animal were really used to have crab, so maybe they are even more, they even more prefer crab over other. But in the literature, what they saw in general is that they tend to prefer crab, then mussel, and then shrimp is really on the bottom of their food preference. And so on the top of that, we also observed that punctually we will introduce them live prey inside the dome just to see during their training how they are behaving. And we will see that motion will really stimulate them, and they will tend to use more arm. And because we want to look at arm recruitment, it might be interesting to combine this contact chemoreception that we observe as well as motion. Overall, we had a general bias in our experimental setup. So first, the food could be retrieved out, and the object could, and the non-prey object could not be retrieved out. So that might have created a bias also. And the fact that we always isolated them to the left also create a bias on the way they use their arms. So what we suggest is to review this experimental setup and maybe randomize these approach of the DOM. So sometime I isolate them on the left, sometime in the right, sometime in the middle, and maybe we'll see if this pattern that we observe right now hold up. Also, in this recording, what we observed is that there were exploratory behavior that we did not score in this study. And Beidel and al. Actually considered the exploratory behavior as their own type of recruitment, and she considered that as more as a sequential kind of recruitment. So further study might include exploratory behavior as a recruitment. And as always, to increase the robustness of our data, it is essential to compare that to field observation. So overall, this study emphasized how important it is to combine sensory system for accurate arm control. Yet, it was interesting to see that arm recruitment and interaction strategies were similar to the one that you observed in the studies that involve vision, and that's open really important perspective on how autonomous an arm is compared to the to the central nervous system. Yet we did not see any arm partitioning when you looked at the overall assay, and that also provide valuable info for behavioral ecologists and also soft robotic engineer on how an octopus will adapt its arm from the first time he made contact, and then overall when he continue further to manipulate this object. I would like to thank my supervisor for their support, their time, and for the transmission of all their precious knowledge, as well as my lovely team for these past 6 months together. Thank you for your attention. I'm gonna put
I believe we have.
here.
Wait.
Can you still see my screen?
Just hold on, Camille. Does it work now? Yes. Thank you, Tim. So Camille, you're still here. Yes. You want us to see your screen, you told me. Yes.
Yeah, maybe I could share it in the PowerPoint so that I can see you at the same time you ask the question.
Yeah. Do you? I think you have to move a little bit your windows so that you can see us.
Let's see.
But if you want, you can unshare your screen and we'll see if we need to come back on your slides.
I don't know if now
So I will propose maybe Laetitia to start with the questions, if you want. So Laetitia, you've been part of the reviewers, and so that's why you're here to ask questions.
Yeah, so can you hear me?
The sound is really weak. Maybe if you can try to...
Can you hear me?
Yes.
Okay, I'm the maximum volume, microphone volume. Okay, so thank you for your presentation. It was very clear, and I really enjoyed reading your work. And I have some questions. So first of all, I wanted to ask you if you discussed briefly about it, but I noticed that you did not talk about the possible influence of the order representation of your stimuli, like during the, let's say, habituation phase, like if the crab was coming in the morning and the rock in the afternoon, or if you switch it, because this might have influenced the let's say responsiveness to the second phases of the test. So I wonder if you took this into account.
So for the habituation phase, maybe I was not clear enough. So habituation phase, they were really not exposed to the non-prey at all. they were only exposed to the food. So during the habituation phase, we really habituate them to the food so that they are familiar with the food and they associate the dome with food, but we never expose them to the object.
Yeah, no, sorry. Then I meant the second phase where you subject them to rocks with agarose infused and crabs.
Can you then repeat your question, please?
Yeah, the question is if you alternated the morning afternoon switches between, if you switch between morning afternoon, or they were consistently done food in the morning and rocks in the afternoon.
Oh, okay. So no, it was randomized. It was tricky to highlight in the slide. So it was either food or object during this experimental phase, but it was really randomized. So we did it with a flip a coin, and so we just like figure whether we will give them rock in the morning and or food in the afternoon. But it was really randomized, and also not to something that I forgot to mention. To avoid, because we know that they're really sensitive to the conspecific odor, they have each of them have their own dome, and if we did not have enough dome, well, usually we'll test per week 2 octopus, so they will have enough dome for each of them, but otherwise we'll soak this in fresh water and then in salt water again to disinfect, so they were also not influenced to the smell of the conspecific.
Okay, another question is always about the possibility that the animal in a way learn what's coming into the dome. If you measure an increasing responsiveness of the animal through the trials, so if you saw that by the end of the week or the end of the second week, the animal would react faster and maybe even insert the same arm into the dome or not.
So it was really individual dependent, but what I saw is that so our 2 youngest in the cohort will increase their responsiveness, but what I saw, the other were pretty old, they were 19 months, and I saw that some of them were not going that often to the dome. At the end of the trial, they were losing interest, and I think it's because of age, because I read a paper where they were saying that with age, maybe they will lose interest when like the task is complexified and it's not as easy to retrieve food. They will not, and we also observe it with our individual when we feed them, they're really like, they become lazy. They want, if the task is complexified. So we did see for sure an increase in the response for the young cohort, but the one that were becoming older, some of them are still responding, but by the end of the trial, the second week with the crab infused, some of them will not come as often to the dome. They will still interact, but after 2 or 3 approach to the dome, they won't come anymore within this 10 minutes that we record.
Okay, yeah, and then with this comment, you come to my next question, which is the pretty large age and size variability in the animals, which is something that normally you kind of avoid. I mean, I perfectly understand that when you work with animals, you had to do it with what you have, especially if they are wild-caught animals. But if you have an animal that it's really small and you do not adapt to your dome in terms of size, wouldn't you expect to have a difference in the number of arms that he insert and also in the identity of the arms, because he would catch the dome in a different way? So
yeah,
did you
especially the small.
Sorry?
The small, especially, they were able. We wanted to review the dome later because the hole were too big, so they were able to put multiple arm at the same time.
Yeah.
So, well, scoring wise, it was difficult for us to figure, but also it created a bias because you really have to go second by second to see which arm is contacting, and so it also bring a bias in our definition of recruitment because you're like you don't know exactly which one was actually first and which one is communicating with this one. So it's really a great point. And I think for further studies, we should reduce the dome or have more homogeneous individual.
Okay. And one other question is about the, I mean, the fact, I mean, you mentioned that the one possibility is that the arm interact with the object that they cannot get through simply because they are curious, and then they interact more, right? So then in this sense, it's obvious that you have many more assays of the non-prey versus the prey. So this is, of course, for me at least, it was really obvious. So I also wondered why it was done, but then you explained why. Now, the thing is that why didn't you measure the time of the first approach?
So we do have it in our data set. I did not mention it in my report, but I do have the time and the first approach. I have the time they take to go as a first approach. And it was really obvious that for food, they approach. And it's true that I should probably have mentioned like the average time they approach for food, especially that I discussed that in my report, that they will tend to approach faster for food. But yeah, they do approach way faster when they detect food.
Well, the thing is exactly this. I do not expect them that they would approach faster if it's food because they don't see it. And if they do that, that means that they use distance chronoception. So that's the thing. Measuring the time by which they put the arm in and they retrieve it, it's exactly, in my opinion, a very good way to exclude that you do have distant chemoception.
Okay.
Because in theory, it should be exactly the same, especially if you do not change the position of the object within the dome, which you didn't.
Thank you a lot, Laetitia. I'm sorry, I have to go ahead with the next but thank you a lot, Laetitia. Maybe we can come back if there is time for more questions. As you arrived only for this presentation, just introduce Laetitia Zulo, who is researcher on octopus, the crossroad between life science and engineering at the IIT now in Genoa.
Yes.
So I will give the floor to Aldine and then maybe go back to Eric.
So thank you. Sorry if it took me some time.
No, you're a specialist, really.
Hi, Camille. Thanks for this presentation. So, yeah, I really curiosity question. So, because you were talking about this chemo, I mean, the fact that octopus, they may have, so sucker that sense and the other one that can test. So in the species you are looking at, so do you know already if it's equivalent in all the arm of the octopus? And can you correlate already with your data?
If like this perception of sense is equivalent between arms?
Yes.
So I do not know between arms, but I know that on the different section of the arm, they might be more able to detect when it's closer to the mouth, so proximal part rather than the distal. But that's like more like belief that and an assumption that you have from me talking to the researcher and the people that I met through my thesis. But I'm not really sure there is something published yet on that, so I don't want to say too much. But I'm not sure that there is. Maybe Laetitia could emphasize if there is, but I'm not sure. You have between arms. It would make sense to compare the connection that we know neurally between arms with that, but I do not know.
Okay, thanks. And do you think that before touching what there is in the other aquarium, so the octopus can really feel already, I mean, test already that there is prey in the water?
So I think there is for sure. For the crab, I don't know because it's a hard shell. So I don't know if the crab extract for the prey will be spread. But what I'm almost convinced, but still it's an assumption, is that on the rock shape that we rub with shrimp, every time you put it in the dome, you like visually see that particles are going away. So I'm pretty sure that it's like, although it's diluted in the water, I'm pretty sure they can detect it and that it's influencing the way they approach and why they come.
Okay. Okay, thanks. And last question, did you really observe differential behavior regarding the character? I mean, from one day to the other, so how the octopus react when you use them already? I mean, several times, you know.
I do observe, so I'm pretty sure my supervisor is watching, so I'm not going to use the word personality because it's really biased. But you can really see characteristic between individual, but the individual itself, because some individuals, someday they will be more stressed, someday they won't like to approach the dawn, so you'll have failures. Someday you'll have to be more patient. So most of them were, every time you put them, because a stress point is that we don't do the experiment in their home. We do the experiment in a new tank. So every time you need to transfer them, and that's a supplement stress for the animal, and not all of them respond the same way. And so some of them, although we allow them like a 10 minutes most of the time, some of them you have to wait 30 minutes for them, like to really see them going out of their den and starting to explore the new dome. So yeah, you do see that. And you have some that will be always stressed, but you have some that on the first week will do really great, and in the second week they will become more stressed. And that's also why on the second week we choose to test only 8, because one of them was that stressed that he was hurting himself, so we prefer not to test him. And so we couldn't use other animal because not other were available, either senescing or with lesion. So lesion is a supplement stress, so we couldn't use them. So we choose to have different number of individuals between 2 weeks.
Okay, and so from the same individual, if you perform the same experiment one day and then the day before, do you get different results?
Yes, that's what it makes it. It makes it difficult. So, and that's also why we choose to use what we saw in this literature, not only for octopus, this anterior and posterior division rather than arm, because for the first time making contact, especially, they will never, like I never see it, use the same arm to initiate contact. Never. They will use anterior or posterior arm consistently. So for example, let's say we have individual 1, he contacted with L1 on the first trial and on L2 on the second trial. So those are anterior arm, but they're never the same. And that was like the same for everyone.
Okay, thank you.
Thank you.
Okay, so we now have a little bit an idea of why you had 9 and then 8 octopus. I give the floor to Eric, now coming back after to Sarah.
Thanks, Camille. Very, very interesting presentation, even if I got a little bit tangled up with all the different arms of the octopus. But, very nice. I also have very curiosity questions, because that's really not my field. I was wondering if this blind exploration, is that innate, or is it something that a baby octopus sort of gets trained on, or through success? Do you know that?
So you mean like something that happened naturally in a... Yes, it is.
Okay,
it is.
So like a newborn, it starts exploring all the little holes it can find.
Yes.
Okay.
I'm pretty sure you have all their senses that plays a key role in that. So if they sense something in their environment, they will go more toward this crevice, this cliff, rather than another. I'm not sure that they will like go in every single hole that they see in nature. But these video that I show you at first, we don't see the octopus retrieving food from him. So it's like also a consistent proof that they do explore, not necessarily to get food. They explore, and you don't know really why, but yeah.
Probably for food, but okay. The other question I had is coming back to the answer you gave to Laetitia about that you saw some of the older octopus taking more time to go to the dome and explore. And so this is interesting, I think, because one of the aging hallmarks is the reduction of nutrient sensing in general. So that's, I think it's interesting. And the other aspect that I was wondering is that do you see, because you said that some of the older octopus were slower versus the other ones were still normal, is it a sex difference between males and females, or is it independent, or don't you know?
I think there is. The only thing is that for some of them it's really obvious to discriminate the sex, but most of the sex discrimination occur post-mortem, and our animal were still like they're still alive and they're still used. We are pretty sure that out of the 9 total animal that we use, 8 of them were female, and we are doubtful on one. Because you have these and even my supervisor looking at them was not really sure. It's really obvious they have on the proximal part really big sucker that will differentiate them and make them male, or they will have these reproductive arm, the ectocotylus, on the third arm, and it's a bit more curved, and so you'll see it. And that's why when we were recording the assay, one of the octopus was rarely using this arm to make contact and more like to explore slightly but not contacting, and we really see this curvature and we assume that this one might be a male, but all of them are really strongly we do think they are female.
Okay, so if most of them are females, then you don't think that there's a sort of a gender bias
No, we do see this age bias though, because when we did the test for the statistical test for inter-individual differences, it was always octopus 8 and 9 were the most different from others, and those one were the juveniles. So you do see this age difference.
And then a quick question: if you get octopus from the wild, how do you determine the age?
It's size-wise, so it's really an estimation.
Okay, but the size depends on how much food they took, right? Got.
Yes, but I believe they have a range of size where they will estimate that this octopus is that month or that month, and that's what I've been told is you look at the size approximately. The one that were reared in the laboratory, the hatchling time, so it's easy to determine, but the one from the wild, it's trickier.
Okay, thanks a lot.
Thank you, Eric.
We have to move on. Thanks a lot, Camille.
Thank you, Eric. Sarah, and we'll finish with
Thank
Stefano.
you very much, Camille. Just one question. So could you come back on your perspective where you introduced the fact that the system, the nervous system of octopus can impact probably the use and manipulation of objects. So could you just present us what are the CNS and PNS system and how you would expect that they have an effect also on the use of arms, or how you think that we could test also this effect?
Okay. So it has been shown in many studies that for sure octopus have a degree of freedom with their arm, and they do not need the CNS, which is the central nervous system, so their brain, to control their arm, and they can perform pretty complex tasks only using information from their arm. And so the PNS is the peripheral nervous system. And so they can use the information from their arm for really complex tasks like fetching an arm, elongating it. They do not necessarily need their brain to do so. And how do they do that? Like, how do they evidence why I don't need my brain? Mostly comes by a lot of study include discrimination of the sense. So some will be blind, like we did now. Some they will give them different shape or different texture, roughness, soft, and they will eliminate either chemo variable, either so it's really playing with the sense. So you have many studies that will do vision chemo, then vision only, then and so play with multisensory studies to figure that out. Does it answer your question? Do you have? Okay.
Thank you.
Well, thank you, Sarah. So we are moving down to Stefano.
Thank you, Camille, for your very clear, nice presentation. It's totally not my domain either, so I have just curiosity questions now that I know what happened to the 9th octopus in the second week. So for example, the environment, you said that the ninth octopus was too stressed, and there is the question of the stress of these animals that you, so the environmental conditions where you keep them, the aquarium where you keep them, and how you move it, temperature, salinity, the current, do you think they may influence this so-called stereotype behavior on the blind
Okay, so for
on your
sure,
experiments?
so for sure, the transfer itself from their home to the experimental tank might generate stress, but we are really lucky to have facilities that have open seawater system, so it's always recycled, but there is a really careful control. So whether you are in winter or summer, you have like the temperature is always the same. It ranges from 15.6 to 15.8, and it's always the same year round. And pH pH is always the same also at plus or minus 0.2. But so those physical chemical parameters are maintained. What I do think, yeah.
So for how long were they in captivity? Maybe you said that before starting the set of experiments.
So the one that were white collected. So we got them. They were in captivity for a bit more than 2 months, and then we tested them. But the other one, they don't have this captivity bias because they hatched in the laboratory. One physical chemical parameter that I do think about is that we put for our experiment setup, we put a curtain that was hiding the natural light. Well, natural light was coming through for us to record, but for them, they did not have any light. And that might be also stress that I just thought about, because when they are in the home tank and you record it, they have the light from the window and they are not covered. So that might be one supplement stress factor. But regarding most of physical chemical parameter, they're so closely monitored that I don't think it might have created a bias. Maybe for the white collected one, if they were not for so long.
So, and also a stupid question about the setup of the experiment. In the dome where you put your rock or your trace, do you put it in a random positions, or is it at the center? Do you think it may have it's always at the center? Okay, so it's always at the same position. And do you think that repeating with higher frequency the experiment, it would change the behavior? It would stress them too much? So maybe I miss it, but did you repeat with the same individual the experiment? I don't know, the distance of I don't know, 1 hour. I'm just making it up.
So yeah, that's true. That's true. Maybe it will be more relevant to test new individual, because we don't know. Well, the one that are currently being repeated, the experiment, are the one with look at motion, to look when you put a live prey. That's the one that will be repeated with the same individual as the one that I tested. But that's for sure that the experiment that I did right now, it might be more relevant to use a new animal that will undergo the same kind of stress rather than use the same one, and maybe we won't have the same result because there will be more stress and also age more. So maybe, yeah, that's true. That's a good point. We should probably use other individuals, which we don't have currently. So that's also why we need to adapt to what we have.
Thank you. I'm sorry. I really have to conclude, but I can propose you to continue talking, and also I'm sure Laetitia, you can continue to exchange with Camille. Just that we are exceeding time here, and so I would like just to conclude officially the session. And before I let you continue to discuss, I would like just to show you, Camille, some message from your team also, we'll say. So here I let you have a look. Yeah, you have a lot, so you say, oh, a lot. Mathilde, Ashley.
Oh.
We have a couple of more. So thank you a lot. You're not here, but I think we can congratulate you again.
Thank you. And you can contact me, all of you, if you have like other more specific question. I'll be happy.
Yeah, okay. And so for everyone, as yesterday, maybe you have time to practice for tomorrow if you still are stressed, or go to the beach if you're in a holiday now. Tomorrow we've got a big day, because so the morning will be with 2 sessions in parallel. We will be in this room from early in the morning, 9. Yeah, confirmed 9, for the session on social science. So really the crossroad between life science and people. And in parallel, starting at 10, yes, we will be also in the other room, Salon de Musique, for ocean literacy and education, with our Blue managers. So you can be here on site, or if you want, you can also be with us, even if the Blue manager students will be online. Okay, so we will let you have lunch when you want and where you want, and we will meet again, but I will explain that tomorrow morning. We'll meet again then for the Pint of Science, the year 1 video pitch in a pub with a drink, to have this social experience as well. Okay, thanks a lot for your participation, to stay in this warm environment, listening to this fantastic science of our MARRES students. Thank you again, Eric, Stefano, Laetitia, also to pass by and to bring your expertise in octopus. Thank you, Sarah and Adine, for the questions. Thank you for the team, also for the constant support. And Roger, we have more hello. And I think you know this person for quite a while. Roger, if you're listening, you have my regards. See you soon. And so I let you find a way to exchange outside of the YouTube live. I think it's better. Okay.
Sure. Thank you, everyone. Have a good day.
Bye. Bye-bye.
Bye bye.